Okay, so you have your DNA, which is transcribed into RNA. And DNA and RNA are in the same language, right? They both use nucleotides. There's only a slight difference. Uracil instead of thymine. And so a transcription is just that. You're just taking one, you're taking a document or something and transcribing it into another document. Or you're saying something and writing it down. It's the same language though, right? The second part, RNA to protein, is translation. And so here you're going from a nucleotide language, which is coding then for protein. And what are proteins made out of? Amino acids, right? So you're changing languages. You're using this template to make something totally different. Okay, so that's the easiest, or that's the functional way to remember it. It's also in alphabetical order, so the C comes before the L. Transcription comes before translation. If that helps. Okay, but then this is the central dogma, so this is how proteins are made. This is how functional parts of your body or cell or everything works. If it's in the DNA, it has the code to make the RNA, which is the code to make the protein. So we're going to talk about transcription. There are 3 stages: initiation, elongation, termination. These are the same stages, general stages used in translation as well, but there's different things that happen. So for transcription, or the initiation, what you have, first a promoter sequence. No, I didn't. Is recognized by what are called transcription factors. And RNA polymerase. And this is upstream of the start site. And is directional. Okay? So it's only on one strand of the DNA. It gears the transcription factors in the right direction so that then RNA polymerase is positioned at the start site and is now ready to start adding nucleotides. So elongation then is the adding of nucleotides. And it happens in the 5' to 3' direction. And what has to happen first is RNA polymerase has to open the DNA. Okay, so what bonds does it have to break in order to open the DNA or separate the strands? Yeah, so it has to break those hydrogen bonds, and it forms a transcription bubble. Where it has opened up the DNA and then starts adding nucleotides onto the 3' end. But the transcription bubble, we don't want to permanently open the DNA and keep it separated. We just want to open it for a little bit, find the sequence that's important to making our messenger RNA, and then close it behind it. So this bubble zips along, but closes behind it as it makes the messenger RNA. Okay, and then it continues to do that until it comes to a hairpin loop. All right, and this happens only in prokaryotes, but a hairpin loop is where the tail of the messenger RNA forms this loop where hydrogen bonds form between the nucleotides, or between the bases and the nucleotides. Okay, and that interacts with the termination sequence, and then the RNA polymerase and the messenger RNA break off and away from the DNA. And the transcription bubble closes. Okay, so at the end of this, you have a string of messenger RNA, single-stranded, and then the RNA polymerase can go back and start another round of transcription. Okay, so what does this look like? It's a nice little video which shows a little more detail, but gives you a good visual of what's going on. We have our messenger RNA, but it's not quite ready for translation. So there's a few modifications that have to happen. So first, you have a 5' cap, which is put on it, and a 3' poly-A tail. So poly-A means there's lots of adenines on the ends, has lots of A's on it. Okay, and these allow it to interact with other enzymes and proteins later. Okay, but it's still not done, so we still are going to make some more modifications. And one of the structures that does that is called the spliceosome. Okay, and this is done by single nucleotide I can't remember actually what that stands for, but SNRPs is what it's called. Random nucleotide proteins or something like that. That plus other proteins make up this spliceosome, and what it does is it cuts up the messenger RNA, discards some of it, and uses other pieces. So if we were to cut this up into 4 different pieces, like this, the spliceosome may then modify it. It would make it smaller, still have its tail and its cap, but then it would only have some of the sections that it originally had. So the ones that it keeps are called exons. And then the one that it leaves out, or the ones that it leaves out, are called introns. So it cut B out and it kept A, C, and D. Okay? But the spliceosome can also have alternate forms of expression in the exons. So this might be one combination, but it may also have another protein it's making where it switches up the exons. Okay, so if it puts D in front of C, then it may make a different protein than if it puts C in front of D. And all other different combinations of introns and exons. And so this allows one gene to code for many different proteins. And it was originally thought that every gene or every DNA sequence that was transcribed had its just coded for one protein. And so they thought there were a lot more genes responsible for the different proteins in the body. But once they discovered this, they realized that, oh, you could make actually lots of different proteins from the same DNA sequence just by switching around the introns, or the exons, excuse me. Okay, so now the messenger RNA is ready for our next step in making proteins, which is translation.